Go ahead.
Hello everybody, my name is Gulsen and today I'm going to present you my PhD project. The title is Assembly Dynamics of Intermediate Filaments, and I will specifically talk about how intermediate filaments elongate themselves. I would like to start by shortly introducing the cytoskeletal components in cells. As most of you are familiar, there are three components in the cytoskeleton: microfilaments, I'll just use the air. Microfilaments, microtubules, and intermediate filaments. So, although microfilaments and microtubules are very similar in their structure, intermediate filaments are quite different. First of all, both microfilaments and microtubules have globular subunits. On the other hand, intermediate filaments have rod-like shaped subunits. And second, while microfilaments and microtubules have polar structures, meaning that their two ends are very different from each other, intermediate filaments have non-polar structures, meaning that their ends are the same with each other. And finally, both microfilaments and microtubules undergo N-mediated assembly of their globular structures, meaning that the subunits can only assemble or disassemble from their ends. On the other hand, intermediate filaments are not very well studied in this aspect. So whether subunits are assembled through their ends or whether they can intercalate along the length of the filament is not known. Also, another thing that is not known is whether they can elongate by fusing with each other through their ends. Therefore, I've decided to study these two hypotheses. The first one is intermediate filaments elongate by end-to-end annealing, and my second hypothesis was that intermediate filaments can exchange subunits along their lengths. But today, in this presentation, I'm only going to focus on the first hypothesis, and I'll try to answer whether intermediate filaments can get longer by fusing through their ends. So before I start showing you my results, I would like to tell you more about my general strategy. I used SW13 cells as my model system, and these cells have endogenous intermediate filaments, although they form very dense networks in cells, and in this situation it's very hard to study individual filaments and follow up their dynamics. Therefore, I decided to work on SW13 cells where they don't have their endogenous intermediate filament network, so it's a perfect host system for me to exogenously express different kinds of intermediate filaments that were tagged to different kinds of colors, like EGFP, which is a green fluorescent protein, or M-cherry, which is a red fluorescent protein. So by fusing these to my proteins, I could follow them in the cell because they are now colored, and also because I can control their expression levels, I could make them less dense and be able to actually resolve single filaments. So in order to answer my first question, can intermediate filaments anneal end-to-end, I used cell fusion strategy. Here I started with two groups of cell populations where I had red filaments in one group of cells and green filaments in the other group of cells, and then I fused them together where I can now have red and green filaments in the same cell. So, and within 5 hours I looked for chimeric filaments in these cells. Chimeric filament means that a filament has an alternating red and green segment. And here's an example of a real cell imaged by fluorescence microscopy, and here you can see in the overlay that I have cells with only red and only green fluorescence, and they are all mixed with each other, and I can actually resolve single filaments. If I look very closely to these cells, I could actually see many chimeric filaments that have alternating red and green segments. Here in the first one, you can see one with 2 green and 1 red segment, and another one is a very long red segment and 1 short green segment. In some rare instances, I also observed circular filaments, suggesting that one filament was able to actually fuse through their ends. So these results suggest that intermediate filaments can fuse with each other, and then by fusing they actually get longer. So in conclusion, intermediate filaments can elongate by end-to-end annealing in cells, and second, fusion proteins that actually make me see those filaments in the cells do not interfere with intermediate filament dynamics. And the third conclusion is that end-to-end annealing is a very common event in these cells, and it's a way for intermediate filament elongation. I would like to acknowledge my PI, Dr. Anthony Brown, and lab members Atsuko, Lina, Paula, and also former lab members Nael, Miraj, Yantin, and Kitty, who